Hello, everyone. You've made your way to the monthly psychedelic support speaker series, and we're glad that you're here. I've been excited about hosting Dr. Matthew Baggott for several months now because I've been following his work with MDMA since the early 2000s. Hard to believe it's been almost 25 years, but Dr. Baggott is a leading expert on entactogens, and we're going to be hearing today about MDMA, how it heals or harms the basic science, which is very timely given that the FDA is going to be making an announcement on August 11th, potentially before, about the Lykos Therapeutics MDMA application for using this as a treatment for post-traumatic stress disorder. I'll introduce our speaker here, but before I do welcome you all to say hello in the chat. You can exchange names and information as we go today. We are recording this presentation and it'll be available on our YouTube channel or on this link that I've already dropped there. If you want to catch up on the video after, we send it in an email follow-up with any of the resources shared today as well. Also, we will probably have time at the end for some questions. You can drop those into the Q&A part of the dashboard. And a note about the chat box, you can toggle between everyone if you want everybody to see your message, or if you just want to message the host or panelists, you can toggle the button there to do that. So let's get to our special guest today. Dr. Matthew Baggott is a neuroscientist who's worked for 3 decades to legally study MDMA and related molecules within the complex regulatory system that surrounds controlled substances. Dr. Baggott is co-founder and CEO of Entactogen, a public benefit corporation that is developing the next generation of MDMA-like medicines. And before starting at Entactogen, he was most recently a director of data science and engineering at Genentech. And I will put the links here to Entactogen and to Dr. Baggott's LinkedIn profile if you want to connect later on and learn more about his work. All right, Matt, off to you.
Hello, everyone. It's great to be here. And so, as Ellie said, I'm going to talk a bit about some of the basic science surrounding MDMA and related molecules. I am not a therapist, and so my ability to talk about healing and harm and things like that is really based on what we know scientifically. And so it may lack some of the nuance that we might hope for from someone who's a very sophisticated therapist, but we'll also get some hopefully useful information and background in how to think about these molecules. So I've been involved in studying MDMA and related compounds really since the 1980s when I became aware of them as an undergraduate. And I worked in what turned out to be the first lab to study the potential toxicity of MDMA and related compounds. Since then, I've gone on to get a PhD in neuroscience, study MDMA and MDA in healthy volunteers, and also continue to try and understand how these drugs work using cells and animal models and things like that. And we'll go into a bit of those studies as we go along. As Ali mentioned, I have some disclosures. I am CEO and co-founder of Entactogen, and we are working on developing new molecules that we hope to ultimately get permission to market as medicines. I also advise the Noetic Fund, which makes investments in healthcare type companies, including psychedelic companies. And I advise Journey Clinical, which helps therapists provide currently ketamine therapy, but hopefully in the future, psychedelic therapy to their existing clients. I'm going to start by talking a little bit about the recent MDMA clinical trials and some of the controversies. So MDMA is currently being reviewed by FDA as a potential prescription medicine. As we've just heard, a response is expected by mid-August. The kind of theoretical deadline is August 11th, but I think that's a Sunday. And so it's likely that the sponsor, what used to be called MAPS PBC and is now called Lykos, will hear from FDA a bit earlier. And there's been a lot of excitement about this, but also there's been increasing controversy. Um, and the excitement comes from the results of the trials, which are really quite impressive. So in the first of two phase 3 trials, and phase 3 trials are the final trials that are designed to show that the medicine or the treatment can benefit patients. In the first trial, we got very nice results. And then a couple years later, we got a replication that had almost the exact same results. They're both really good. And we saw that the patients who received just placebo medicine and psychotherapy improved, but the patients who received MDMA and psychotherapy improved even more. And that's a consistent finding in both of these phase 3 trials. In addition, although we don't have full long-term results from these trials, looking back at the earlier phase 2 trials, it does look likely that these benefits are persisting, which is very good and very promising for the potential for these medicines to help people. But there have been increasing criticisms about the trials. And if we look at the categories of criticism, you can kind of sort them into two areas. One is how the studies were designed, and the other is how the studies were conducted. There is a really rich discourse here, so I couldn't possibly summarize all of the topics that have been discussed, but just some of the example issues that have been raised include the fact that the study design made it really easy to guess who received MDMA and who didn't if you were around those people. There was no real effective blinding. And so the concern here is that if researchers become aware of who's gotten the MDMA and who hasn't, they might maybe not be as enthusiastic about taking care of and providing therapy to those who didn't, just because they might have some idea in the back of their head that those people are less likely to do well. So you could have some kind of unintentional biases forming like that. Similarly, in the participants, those who didn't receive the MDMA might become despondent. They might have an anti-therapeutic effect of that. They might be more likely to drop out, and those who receive the MDMA may be more excited and may work harder to progress and things like that. And so there's some concerns there. Additionally, the main outcome measure for the study, what's called the CAPS-5, which is a structured interview that's performed by somebody, in this case, who's not closely involved in the study and wouldn't have known who got MDMA or who didn't. This questionnaire goes through people's experiences with the severity of their PTSD symptoms, but it focuses down on a single trauma and asks really about that one trauma. However, a lot of the participants in the study had multiple traumas often, and so it raises the possibility that people could have appeared to have gotten better on this measure because they were worried less or concerned less with their index trauma, while not actually really having much relief of their symptoms and issues because they had other things that became more prominent. In terms of how the studies were carried out, some of the concerns are that personnel and participants were often people that were kind of from the psychedelic community, really thought the MDMA was going to work, and thought that participating was a way that they could help get the drug approved. And that's atypical for drug trials. But on the other hand, if you go back to when these trials started, there wasn't really much of an expectation that this was going to work. It was kind of considered a quixotic thing for Rick Doblin to spend all of his time trying to get MDMA approved as a medicine. And so, of course, the people that ended up helping would have been people that were disproportionately inclined to believe that this might be useful. Another concern has been that adverse events, well, before I move on, what you would hope, though, is that as the work matured, that you would then start to bring on more professional drug development and clinical trial expertise that would then end up kind of ensuring that the existence of optimism in this treatment wouldn't end up causing any issues. The other concern that's been expressed is that adverse events may have been observed or mentioned by participants, but may not have ever been recorded in the formal measurements. And this can be an issue in studies where if somebody says, hey, I've been feeling this way, and then you get them to fill out a questionnaire the next day and they don't mention it, it doesn't go recorded. And so I can't adjudicate the extent to which these any adverse events were not recorded or were recorded. What we can do is look at some of the things that might have influenced the overall outcome of the trials. And we'll get back to that in a second. So in terms of how the studies were designed, it's important to note that they were designed in consultation with FDA. So there was this process called a special protocol assessment where essentially the FDA signed off on the design of the trial. And because of this, it's unlikely that these issues with people being able to guess who got MDMA or the limitations of the CAPS-5 measure, it's unlikely that those will end up affecting whether or not the treatment gets approved. However, these types of concerns may affect the willingness of payers to provide coverage for the treatment. With regard to how the studies are carried out, these are things that could affect the possibility of FDA approval. My take is that these would probably need to be so widespread and pervasive that they actually affect the CAPS-5 scores, the primary outcome measure. And one way to get a sense as to how likely this is to look at the retention rates in the study. And so here's retention from the second phase 3 trial. And so we have the different time points at which important outcome measures were made. And then we have a column for the number of MDMA treated participants who are still in the study and providing data, and the placebo treated participants who are still in the study and providing data. And generally, there's not a lot of people who left the study, particularly those who received MDMA. And this is reassuring because a way that you can end up with unreliable results is if the people who aren't doing well decide to drop out of the study. And so the fact that only one person in the MDMA group dropped out gives us some reassurance that the results should be reliable. And similarly, in the placebo group, you might also expect that people who are dropping out there might be people who are not doing as well. But if those people did particularly poorly, it would have only heightened the difference between the MDMA and the placebo treatment group. So this is overall reassuring that at least this trial is likely to have reliable data for the primary outcome measures, as long as there wasn't something really systematic going wrong, which we can imagine that FDA has a lot of expertise at evaluating, and hopefully they will evaluate it, and in the end, we'll learn the truth. So MDMA does appear to have significant benefits looking at the data at face value. It would be reassuring to many people if there were data from some organization other than MAPS and Lycos. And thankfully, there are starting to be trials in Australia that are giving MDMA for people with post-traumatic stress disorder. And so we may have some independent confirmation of this overall effect. It would be nice to have this confirmation because MDMA seems to have promise not only for post-traumatic stress disorder, but also for a number of other indications. And it seems in that way like SSRIs, but possibly more efficacious. And so some of the indications that we have, some of the reasons to think that MDMA and other entactogens might be useful include results from an open label trial where MDMA seemed to be useful for alcohol use disorder. In addition, in the phase 3 PTSD trial, if you look at the patients who had problematic drinking, drinking seemed to be improved in those who were treated with MDMA. And similarly, people who had eating disorders in that same trial also seemed to improve. And if you go and start to add up all the different potential issues where MDMA or entactogens have been proposed to be useful, you end up getting to a really impressive percent of the adult population. So if MDMA does end up being useful, it may well be useful for something close to 1 in 4 people. So how is it working if it does work? The first person to suggest that MDMA might be useful was Alexander Shulgin, Sasha Shulgin. When he was later asked why he thought that might be the case, his description focused on what essentially seemed to be like increased honesty with oneself and less avoidance. And so it was as if his ability to solve personal problems was enhanced. In studies where we give MDMA to healthy volunteers, we can measure some of the things that he talked about, as well as other experiences that people have suggested might be helpful. And so in one of the studies I ran, I tried to measure some of these aspects. And just starting on the right here, one of the things that we see is people given MDMA in a controlled setting report feeling more authenticity, meaning they feel more like they can be themselves without having to kind of self-monitor or worry about other people might think about them. Similarly, if we look at the middle plot, people report less social anxiety. So just as a brief note, in these plots, usually the 0 point in the bottom left is when we gave the MDMA or placebo, and then we do repeated measures from the left to right at different time points. And so you see 0, 1.5 hours, 2 hours, 2.5, 24 hours. And then we will typically show the average result with a dot or a triangle or something. And then these up and down bars show the variability, in this case, the standard error of the mean around that response. And so social anxiety is decreased by MDMA compared to placebo. On the far left, there's a nice, fast, easy measure of emotional closeness to others. We just ask people to respond on a 0 to 1 scale by making a mark on a line. And what we see is that people report feeling increased emotional closeness after they take MDMA, and it lasts about 2 hours and then starts to diminish. There would be other ways, of course, to try to measure and think about the acute, potentially therapeutic effects of MDMA. Increased self-compassion seems like, a good way of thinking about it. Also decreased neuroticism and avoidance, or ability to be honest and think about things that normally might upset you. And that last one does seem pretty close to what Sasha was talking about in the previous slide. Just as a side comment, when we do these kinds of studies in healthy volunteers, it doesn't seem like feelings of love or extroversion and wanting to be around other people are super reliable, common experiences. Maybe 60% of people might report that kind of thing. And it does seem to me like it's a little more common to have as a core experience something closer to this authenticity thing, or what Sasha was talking about in terms of increased honesty, or ability to think through things in a calm way. So almost as soon as people started taking MDMA and trying to understand it therapeutically, scientists like Sasha and Dave Nichols started trying to understand its mechanisms, how it works in the brain. And so if we look at a history of this, we can kind of think of it as, starting on the left, people realizing that serotonin is involved. And then as we progress, people started saying, yes, but there's also other things going on. Dopamine seems to be important. The serotonin seems to cause oxytocin to be released. And then we get to the present, where people are back in some ways to focusing on serotonin, but they're doing a much better job using contemporary scientific tools of figuring out which exact brain regions are involved and what might be happening. So one important finding in this history has been the idea that you can look at the relative effects on dopamine transporter, or DAT, and the serotonin transporter, or SERT, and predict whether something is going to be roughly MDMA-like. So here we're going to plot horizontally just whether something is biased to affect the dopamine transporter and increase dopamine extracellularly, or the serotonin transporter and increase serotonin extracellularly. And the things that are more on our left that have lower ratios are the ones that are serotonin biased. And those end up often feeling MDMA-like. Whereas the ones that are to the right on this plot that are more dopamine biased and have these higher numbers, they appear to be classic stimulants. And so this seems like a pretty good rule of thumb for just looking at a molecule's pharmacology and saying, will this feel like an MDMA or not? But it's imperfect because there's a lot of these things in the middle that are often feel, if you give it to a human or if a person takes it, they often have a lot of the MDMA-like feeling, but some of them don't. And so it suggests that there's additional pharmacological mechanisms beyond just the serotonin and dopamine ratio that may be at play. There is a large number of studies out there at this point showing that serotonin really is important for MDMA's effects. And so one that I helped conduct was where we gave MDMA or placebo to healthy volunteers. We showed this standard increased feelings of emotional closeness to people. And then we tried giving an SSRI. If you gave it on its own, you don't really get any effect. But if you give it before the MDMA, then you very much blunt the emotional effects of MDMA. And the idea is that the citalopram, the SSRI, attaches to the serotonin transporter better than MDMA can, and it prevents MDMA from accessing the transporter, which means MDMA can't release serotonin and you don't get the emotional effects of MDMA. It's not obvious why serotonin would be the thing that causes these emotional effects or increased problem-solving abilities. But there has been a recent proposal that serotonin might be important for helping the brain switch between already learned automatic processes and slower, flexible problem-solving processes. Before this paper came out, I typically thought of serotonin as doing something like stabilizing ongoing neural activity. And so you've got these serotonin cells that are down in the brainstem, and then they extend all over the cortex in the brain. And they often fire at kind of a stereotypical, like pulse-like fashion. And it seemed like more serotonin kind of helped stabilize whatever was going on and less made things more likely to switch around. But the idea from these researchers is that serotonin can help engage in a mode that's more problem solving. And serotonin, a lot of it in the body is actually in the gut. And so they suggest that there is a fundamental similarity to the gut serotonin helping digestion and the brain serotonin helping essentially cognitive digestion, or like thinking through new things and processing them. If this is true and useful, it still raises the question of why would serotonin be involved in things like sociability, and maybe, and this is just purely speculation on my part, maybe for mammals, problem solving is something that we've evolved to do together. And so that as you move into a problem solving mode, maybe you also tend to move into a more social mode. So we can measure these brief emotional effects of MDMA that typically around 4 hours are starting to diminish. But there does seem to be interesting MDMA effects that last more hours or even days beyond that. And one of the most intriguing findings in this regard is from Gould Dolan and her colleagues, where they found evidence that MDMA was able to enable a type of social learning that was usually for mice only possible for a brief period of time in their development, but then would become impossible to access, whereas MDMA made the animals able to access this type of learning. And one of the most intriguing aspects of this finding was that there seemed to be a relationship between the duration of the acute effects of MDMA and other drugs and the duration of the openness of this window, how long the window remained open for. And so from a follow-up paper, they visualized for essentially different psychedelics, how long the acute effects of the psychedelic last, and that's at the top of this visualization, and MDMA is shown kind of in the middle in red, and how long this window remained open, this ability to learn that normally was hampered in adults. And it does seem like there's a rough relationship here. A caveat that we should note, though, is that these are mice, and it's a type of learning that it's not clear how it really relates to psychotherapeutic effects in humans. And I think a lot of work is going to need to be done to really start to map this. But it is certainly an appealing idea that MDMA and other substances that have psychedelic-like effects might improve ability to learn for some time period after the experience. And thus people might be able to get do more work, make more therapeutic progress, not just during the experience, but in the hours or days afterwards. In terms of what we do know in people, there are lots of reports, both that people after MDMA sometimes have lowered mood, what's called come down by users, and sometimes also have improved mood, which is called afterglow by people. And so this is visualization of results from a large study where they had over 800 people who had used MDMA, and they asked them, on a scale of not at all to very strongly, do you experience a come down or an afterglow? And you can see the results here. It does seem like, say more than 1 in 4 or close to 1 in 5 people report some kind of come down, and there is a similar amount of people reporting an afterglow, although there's a lot of people who report no afterglow at all. So there may be something here. And starting with this idea of an afterglow, here's just one of the reports that I pulled from Reddit just to give a sense as to how people sometimes talk about that. So this person reports that they did what they consider to be real MDMA, and they had positive afterglow effects that made them less depressed until the 5th day afterwards, when they went back to sort of their normal level of worries. I used to essentially ignore these kinds of reports and say, okay, yeah, after you have positive experiences, you feel positive. But then I started to become aware of ketamine and its use as a fast-acting antidepressant. And I started to wonder, well, maybe this is a signature of a fast-acting antidepressant, and it's just maybe that it only lasts for a few days, but could potentially have similar mechanisms to ketamine. And in fact, there is evidence that MDMA, like ketamine and like classical psychedelics, can increase neuroplasticity. And so here's results from a supplemental part of a paper from David Olson's lab, where Calvin Lee and colleagues exposed neurons to different concentrations of MDMA, which is in the top left corner, or other related psychedelics, and measured how much increase in sprouting of neurons they saw. The vertical axis, this percent efficacy, is a measure of that degree of sprouting normalized to ketamine, which is shown in the top right. And so MDMA does seem like it's behaving like these other things in terms of increasing neuroplasticity in this assay. And what's remarkable here is that MDMA is doing it at a really low concentration, and the overall effect is really quite large. And so to the extent that this is a mechanism of an antidepressant effect or some kind of positive influence on mood and on ability to learn or change, it does seem like MDMA might have similar type of effects as ketamine and classical psychedelics. At Entactogen, we've started looking at some of our novel molecules as potential antidepressants. And there's a measure that's sometimes used called the forced swim test, where animals are first given a drug that might have an antidepressant-like effect, and then they're placed in essentially a bucket with high walls with water in it. And so these rats will go into the water, and then they'll try and find a way out. And rats are really quite smart. So they've never been in one of these things ever before. They've probably never been in water before. But what they quickly do is they figure out, oh, I can't get up the sides. It's too steep and too tall. So maybe there's some way to get out if I swim down. And so they start to swim down and look around for a way out. For some reason, drugs that have an antidepressant-like effect will make the animals search for an exit longer. And so they'll swim more and look around more. And eventually animals give up and just start floating there. They're like, I can't get out. I just need to wait until someone rescues me. So this isn't really a model of depression because the animals aren't depressed, but for some reason it is a signal that we might have an antidepressant-like effect. And so if we give one of our novel benzofurans that has MDMA-like effects to the animals and do this the day after, so after the acute effects of the molecule have resolved, we do see evidence of an antidepressant-like effect. So at the higher dose here, we're seeing less time just floating and more time swimming around looking for a way out. And this isn't something that's just caused by them being non-specifically energized the next day, because other measures of activity aren't really being changed. So there might be something here. And this isn't an idea that we just came up with. Similar results have been published for methylone, the beta ketone analog of MDMA. So there's a reason to think now that there might be something going on with these molecules in terms of an afterglow-like effect in animals. So that's the afterglow. And that seems like it's something that is experienced soon after the molecule's effects wear off and might last for a few days. The other side of things is this lowered mood, this come down, and this typically doesn't initially develop, the first day or the second day, but often is something that people start to feel on the third day. We ran a study to try and see if we could detect a come down and prevent it. And we weren't quite successful at detecting it, but we did have some interesting results. So the theory for this was based on the fact that in animals, if you give a high dose of MDMA that causes a serotonin depletion, so here we've got a black bar going sideways showing brain serotonin levels of an animal given placebo, and then slightly above that in red, we've got a bar showing, going to the left, brain levels of an MDMA-exposed animal, and this is a high dose of MDMA, and the result is that the animals have lower serotonin at 7 days after they got the MDMA. If after giving that kind of dose of MDMA, an SSRI is given 4 hours after the MDMA, so essentially after the animals experienced a lot of the MDMA effects, you prevent a fair amount of that serotonin depletion. And so you end up with this intermediate level for the combination. We thought it would be helpful to try and do this exact same manipulation in healthy volunteers. Of course, with a much lower dose of MDMA, that's the typical therapeutic dose that we don't think causes any kind of serious brain issues, but which may be causing some kind of temporary serotonin depletion or temporary serotonergic changes. So if we do that, and this is the same measure as I showed before, emotional closeness to others, we don't really have any change in the overall time course of the basic MDMA effects. And this isn't too surprising because we're giving it at 3 hours after the MDMA, which means that most of the emotional effects have happened and MDMA is starting to wane in terms of its intensity anyway, although there's still quite high levels in the blood and in the brain. If we do this, what we find is that we protect people against being a little out of it the next day. And so if we give a demanding cognitive learning task, people are given placebo, perform at a certain level, and if you're given MDMA the day before, then you're a little worse. And if you have this combination, it prevents that MDMA-related decrease in performance. So it seems like people given the SSRI are a little less out of it. And I don't show it here, but the SSRI alone doesn't change performance. And so this isn't just an additive effect, but it really is an MDMA effect that's being reduced by the SSRI. We had hoped in this study that we might be able to see evidence of a come down several days after the MDMA. And we specifically recruited people that reported often experiencing these kinds of lowered mood in the days after MDMA. But what ended up happening is we had a lot of missing data after we gave these people MDMA, and we were collecting the data with an automatic process. And so we didn't notice this until quite late. Whereas if we gave the SSRI after the MDMA, it very much decreased the missingness of the data. And so it's hard to interpret that, but it does seem like something where we should follow up and try and run another study and see if we can do a better job of preventing an MDMA-related loss of data. However, there are people out in the world who do this kind of thing. They take SSRIs after the MDMA to try and prevent a come down, and they do seem to have benefit anecdotally. One question, of course, is whether these cognitive impairments really do relate to serotonin or not. Is this really we're preventing a depletion, or is it just some other mechanism? The answer is we don't really know that yet. But what it does seem to be true is that there is a little less tendency to be out of it the next day if you take an SSRI on the point in time when MDMA's effects are decreasing. And anecdotally, we have reports like this that we are not yet able to confirm in our studies, but which I think would be useful to follow up on. So I also wanted to talk a little bit about two other things. One of them is hyponatremia, and the other is the concept of loss of magic or formation of long-term tolerance or reduction in the efficacy of MDMA. So hyponatremia is low salts in the blood. And what seems to happen is that MDMA impairs the body's ability to regulate water balance. This is an issue that really only started to occur after harm reduction efforts began to focus on avoiding dehydration. And so it seems like it's a problem caused by overhydration. But the level of overhydration needed to cause these kinds of problems seems fairly modest. So MDMA-related hyponatremia isn't related to high doses of MDMA. In the literature, it seems like it happens to some people with a half tablet, not particularly high doses. And we have some data that I'll show in a second where we gave 1.5 mg per kg, which is, a little over 100 milligrams. And it also is something that isn't just associated with the peak effects. And sometimes people get into problems with hyponatremia after they have largely come down from the experience. And sometimes you see cases of it the day after MDMA use. When people get into problems, it's almost always women. And the reason for that actually seems to be that estrogens alter some functioning of pumps in the brain that help to pump water out of the brain cells. And so in women, hyponatremia has a greater tendency to cause brain cells to swell and thus stop working as well. And so the kinds of symptoms that you get when this happens are things like nausea and vomiting, headache, confusion, feeling irritable or low energy, muscle weakness or cramps, and in the worst situations, seizures and coma and potentially death. I always try to go out of my way to mention this issue when I talk about MDMA, because when people start to feel bad, like headache or drowsiness or things like that, it's really common for people to think, oh, I wonder if I'm hydrated. Maybe I should have some more water. And that can be really dangerous, particularly for women. And so it's much safer to try to start your experience well hydrated and then be careful to have a limited, though adequate amount of water. And so this can be typically done by filling a water container at the beginning so that you know how much water you've consumed. And also, it may be helpful to have sports drinks, not necessarily with sugar in them, because that doesn't really end up helping, but sports drinks that contain salts that would prevent the fluids you're drinking from causing much hyponatremia. So here's results from one of a pair of studies that my colleagues and I ran, where we gave MDMA, and this is again 1.5 mg per kg. And for some people, we gave placebo, and this is 12 people, 6 male, 6 female. And after an hour, we asked them to drink a fairly large amount of water. So 20 milligrams per kilogram body weight. So for a 70-kilo person, that's the equivalent of about 3 pint glasses. So a large amount in a relatively short period of time, a half hour, but not something that's difficult to drink. Maybe a little difficult, but not an insane amount of water. And what happens if you're on placebo is that you get a drop in your serum sodium and then the body recovers over a few hours. If you're on MDMA, this drop is lower and lasts longer. And this red line that's dotted and goes across horizontally is at 135, which is a standard definition for hyponatremia. Values below that are considered hyponatremic. And so we did induce hyponatremia on average in people. Because it was just below normal, people didn't have any symptoms, but this would be the kind of thing where it shows that a relatively modest amount of water can cause hyponatremia. We don't show on this plot what would happen if people hadn't drunk any water, but we know from other MDMA studies that serum sodium remains relatively flat. So that's one potential safety concern that on the face of it isn't related to particularly wild behavior or particularly high doses, and is still something that people may misdiagnose. The other aspect of MDMA that I wanted to mention was the phenomenon of loss of magic. So repeated MDMA users sometimes report that MDMA has diminishing effects on them. Sasha Shulgin named this phenomenon loss of magic, and he described it as something that he never recovered from, where he went from having these really incredible experiences to having kind of mild stimulant-like properties with annoying side effects. And he never was able to recover and get back to these magic experiences. I was interested in how often this happens and whether it was predictable. And so I tried to collect online data from 600 people and asked them whether the experiences of MDMA had changed since they started using it. And about 40% of people said yes. When I asked follow-up questions, it did seem like it was almost always that the most interesting and rewarding effects of the experience had decreased. I tried to see if I could find something that would predict that, and I asked questions both about the number of times they'd used MDMA and also the intensity of their use and the doses they used. And the thing that seemed most predictive, although not particularly strongly predictive, was the number of exposures. And so here's just a plot where on the horizontal axis, we have the number of times that they reported having used MDMA, and each dot is a person, and I've made them slightly translucent and slightly jittered them so you can kind of see where most of them sit. And then the blue numbers and the diamond is the median value for each of these groups. And so you can see that the people who said no, they don't have changed effects of MDMA have typically used around 8 times, and the people who are unsure have typically used around 8 times, and the people who say yes have typically used around 20 times. So there is a bit of a difference there, but there's a lot of overlap here, suggesting that there are also personal variables that we're not able to measure. We've also tried to look in our studies where we gave MDMA to healthy volunteers to see if this phenomenon exists there. And so this is a study where we had pooled data from my collaborators in San Francisco, Harriet DeWitt's lab in Chicago, and Matthias Lichty's group in Basel. And we mostly weren't able to find strong evidence of this. So each of these dots is a participant, and It's their peak response in each of these measures. And then these lines that are being drawn are lines for each of the 3 study sites. And it was only for any drug effect that we saw what seemed like a statistical effect that might indicate that people with more exposures were having decreased experiences. And so it's kind of a maybe, but because we're combining across these different groups, there's a lot of noise here. If you just eyeball these, it does seem like it's plausibly something going on. So to the extent that we can guess a mechanism, a plausible one is the serotonin transporter being lowered. And this might be a specific lowering of the transporter, but it may be just that the transporter is the easiest way to measure just the overall serotonin functioning in people, because we can use PET or SPECT, and we can take something that sticks to the transporter that has radioactivity so that we can pick it up with an imaging technique. And so there's been enough studies done that have tried to measure the serotonin transporter in MDMA users that you can combine all the studies into a visualization. So this is something that Van de Black and Dumont did. So each point is one group of users from a study. On the horizontal axis, we have how long it's been on average since people in that group last used ecstasy. And then vertically is just their level of serotonin transporter compared to the controls within that study. And if you look at it, it does look kind of like people who've used recently often have lower serotonin transporter, but people who've been abstinent for a long time period have normal levels. And so this suggests there may be recovery with abstinence, which would sort of go against what Sasha seemed to think of permanent loss of magic, but it's possible that this is not quite the same thing as the magic he was talking about. Unfortunately, there's this gap in the data set between around 4 months and around 18 months. And so it's a little hard to know what the intermediate level of recovery might be. And in any case, these are different groups of people, so it's not quite recovery that we're seeing here, although it's possible that that's underlying the phenomenon. So that's some of what we know about MDMA. And I wanted to end by talking a little bit about the work we've been doing to develop newer compounds. And kind of the starting point for that was a compound called 5-MAPB. In the history of understanding and research on MDMA's mechanisms, one of the things that happened relatively recently that sort of surprised me was on the online forum Reddit, somebody announced that they had come up with a cocktail for recreating the MDMA experience to the level that experienced people couldn't distinguish this cocktail from MDMA. And once I read this, it became really interesting to me, and I tried to figure out, well, what can this tell us about how MDMA works? And it turned out that the key ingredient in his cocktail was 5-MAPB, a benzofuran derivative that is structurally quite close to MDMA. And so here's the MDMA structure, and then here's the 5-MAPB structure. And you can see that there, on the left of the molecule as it's drawn here, there's a difference where MDMA has 2 of these red oxygens and 5-MAPB only has 1. And instead there's, a double bond if you're a chemist. But the main thing to think about is that it's missing one of MDMA's oxygens. And this ends up being interesting because the oxygens are a relatively easy way for enzymes to open up that ring and metabolize, break down the MDMA. And so by taking it away, the 5-MAPB ends up being a little slower for the body to metabolize, and it ends up having a longer lasting experience. So we've been collecting data on people's comparisons between these experiences. And what people tend to say is that they both have similar therapeutic emotional type effects, but that MDMA has higher sort of energy or activation, but is also a little more kind of, maybe confusing, or you feel a little more scatterbrained and not as clear. Whereas the 5-MAPB, you have less energy, but you're maybe a little more clear-headed. The 5-MAPB seems to last longer, and people think that it has a milder come down. So this would be promising for use in therapy, if it's true, you potentially wouldn't have to redose and you might have fewer after effects. So we started characterizing 5-MeP-B. One thing that we've been doing is just asking people about their experiences, and we have an online questionnaire that we're continuing to collect data with. And so if you ask people to answer questions about social anxiety and self-compassion, it does seem like both molecules decrease social anxiety and increase self-compassion. And then if you ask about positive emotions, it looks like, consistent with its reputation, 5-MeP-B does produce positive emotions, but they tend to be a little less energetically high states than MDMA. A way that we try to study these molecules legally and reproducibly is with a rodent drug discrimination paradigm. And so we train psychonaut rats to distinguish low doses of MDMA from placebo. So if they're given MDMA, they're asked to press one lever and they learn to do that. And then they get a little tasty reward shown as that little orange ball. And then if on other days when they get the placebo, the vehicle, they are asked to press another lever and they learn that if they do that, they'll get a reward. And then we can give them a novel compound on a test day and see if they want to press the MDMA lever, indicating that they think it's more like MDMA or that they think it's more like the vehicle. And in either case there, they'll end up getting a tasty reward. So we're just purely measuring what their belief is. This is a useful way of studying things that had been used by Dave Nichols and his group and a number of other researchers. One of the reasons I think it's really nice is that rats can tell MDMA from placebo at quite low doses that are very similar to the doses that produce therapeutic effects in humans. Whereas all the studies that you might read about or hear about that look at opening of these critical windows or causing increased sociability or empathy in the animals, those are typically using doses that are 5 or 10 times higher. And so I like to think that these are very cleanly measuring the MDMA effects of greatest interest. A reason that this isn't done that often is that it requires a lot of work to work with the animals and ensure that they're happy and healthy for weeks and months at a time. And so a lot of effort and credit should, a lot of effort comes from and a lot of credit should go to our collaborators, Lisa Baker and Candace Johnson and Rachel Burrows, who are collecting these data. So when we do this kind of thing with 5-MeP-B, and let's just look at the left plot, we can increase doses of 5-MeP-B or MDMA and then see what % of the time they're pressing the MDMA lever. When we do this, we find that 5-MeP-B looks a lot like MDMA. As you increase the dose, the rats are more likely to press the MDMA lever until you're around 1 mg per kg, 1.5 mg per kg, and the rats are 100% pressing the MDMA lever. These 5-MeP, we're labeling 5-MeP-B here with this RS because MDMA and 5-MeP-B are actually a 50-50 mixture of what we call enantiomers. And so there's essentially a left-handedness or a right-handedness to the molecules in their three-dimensional structure, and these can have different pharmacology. So one thing we've done is we've looked at the enantiomers of 5-MeP-B, and we found that they're both active, which is a little surprising because with MDMA, the R is pretty inactive. And so we can look at the DAT-CERT ratios of the two enantiomers, which I have at the bottom here. And I'm starting to speed up a little bit because I'm keeping my eye on time, and I was really hoping we could chat a little bit and I could answer questions. If we look at the DAT-CERT ratios, this thing that predicts whether something will feel like MDMA, MDMA is about a 0.6, and the S and the R enantiomers of 5-MeP-B are, not too far from that. But one thing that we figured out is that you could mix those in non-50-50 mixtures and end up with something very close to MDMA. And so a non-racemic mixture of 5-MeP-B seems to possibly be better than MDMA in some ways. We've additionally looked at modifying the molecule further. So methylone, which is the beta-keto analog of MDMA, is something that's been used therapeutically since the 1990s when Peyton Jacob and Sasha Shulgin invented it. when they patented it in the mid-90s, they suggested that it could be used as a potential antidepressant, although that was partly just because that was a legal way of saying Entactogen, and they were trying to keep the molecule legal. It was eventually made illegal in the early 2010s. These compounds are interesting because they tend to not cause the serotonin depletion or a monoamine depletion, and they also don't seem to produce as much tolerance. And so it's possible that you would get less loss of magic with one of these kinds of molecules. So here's MDMA and methylone. And so you can see what's happened is that methylone is adding on an oxygen on the little right chain of the molecule. And so what we thought is let's make the equivalent version of 5-MAPB, so a beta-keto 5-MAPB. When we do this and we look at the two enantiomers, the left and right-handed molecule, we find that the S enantiomer is MDMA-like and the R is not. And when we look at the cert ratios, S5-MAPB is very similar to MDMA, much more similar than methylone for that matter, suggesting that it may plausibly end up feeling closer to MDMA than methylone does. We've looked at whether this molecule causes depletions when it's given in really quite high doses, and it does not seem like it lowers serotonin 2 weeks afterwards. So these are data showing serotonin levels in the hippocampus, and each dot is an individual animal. Controls are at a certain level if you give a fairly low dose of, well, low but active dose of this molecule 3 times every 2 hours. And we're doing that to kind of imitate the human exposure, because humans will not metabolize and clear drugs as fast as rats. So if you want the rat to have a similar exposure to the molecule, you need to give it repeated doses. But we're in this middle category, not really raising the dose, we're just giving an active dose 3 times. And then in the top bar on the plot, we're increasing the dose to 5 times an active dose and giving that 3 times. And we're not getting any serotonin depletions at 2 weeks. So this seems like a promising molecule in terms of not causing serotonin changes in animals or humans. So those are some of the approaches we've taken in our preliminary results in trying to come up with a next generation of MDMA-like molecule. MDMA has all these issues that, even though it's going to help a lot of people, are going to prevent it from maybe being as fully leveraged and used as we would like. And some of these are things that are going to make it necessary to have clinical monitoring and will increase expenses. And some of these are things that will decrease experiences for patients. And so what we'd like to do is find a way of having these experiences and treatments available to more people. And so the question I'd like to leave you with, and the thing that we really would like to work on with the larger community once we get to that point in drug development, is how could society really use these next generation compounds to support human flourishing? If we can use these medicines outside of clinics, how could we do this best? And how can we ensure that interventions are safe and helpful, and that we're really building community and building closeness and connection with each other as we do it? So thanks for your attention. Sorry there wasn't more time left for questions and things, but feel free to reach out to me. That's my email at the bottom. I'm active on Reddit, and Entactogen is active or semi-active on Instagram and LinkedIn. And so thank you again for your attention and for letting me come here and talk with you.
Awesome. Thank you, Dr. Baggott. I have a lot of questions, but we'll go ahead and get to the audiences first, since we just have a few minutes here. There's a question from Michael who's asking, what are your thoughts about the recent data reflecting MDMA's role in stimulating the 5-HT2B receptor, and how this might potentially influence the valvular issues that are being hypothesized as possible with other classical psychedelics in the heart?
So I do think there's cause for concern in general with molecules that stimulate the 5-HT2B receptor, causing proliferation of cells in the heart valves, which makes the heart valves thicker so they don't open and close as well. And this is something that happened to Sasha Shulgin, and he needed to have surgery to repair his valves. And that's a really intense type of surgery because you're like working with the thing that keeps you alive and you're trying to ensure that you don't lose oxygen to the brain as you're doing it. So it's a scary kind of surgery. I tend to think that it's not going to be an issue with widely spaced exposures to MDMA or related compounds. And also that with things that release serotonin, the serotonin itself is going to stimulate that receptor. And so focusing on whether or not MDMA or a benzofuran stimulates a receptor directly might be beside the point because you're going to get stimulation anyway from the serotonin release.
Okay, great. Thank you. And the follow-up, another question is related to, do you recall what the serum sodium levels were in reports of hyponatremic patients?
I don't.
Perhaps a more relevant question for this audience might be, what are some signs and symptoms if someone might be getting into a risky area of hyponatremia on MDMA?
So I tried to put those on the slide. They're often things that are kind of non-specific, and so may not be obvious, but feeling like confused or sedated, muscle cramps or spasms, agitation, and so they can be difficult to cleanly identify as being related to hyponatremia. As I said, symptoms almost always happen in women. Men, when they get hyponatremic, at least for a few hours, will typically not show any symptoms and will typically recover without problems. So this is mostly a thing we should worry about with women who take MDMA. Similarly, women athletes who are maybe like running marathons and are drinking lots of water can also get into problems with this. And so there's a large literature on this. But yeah, I think the safest, best thing to do is to start out well hydrated and then to have controlled amounts of water during your experience.
Yep, and I'd say that is good across the board for any psychedelic substances to be well hydrated before. Okay, well, I'll ask one more question if you have another minute.
Absolutely.
Okay. This one's related to prolactin release. And do you have any thoughts about any post-dose supplements that may help, either specifically related to prolactin or I'd say the serotonin decreases that have been observed?
That's an interesting question. So there's definitely a history of taking 5-hydroxytryptophan to try and help replenish serotonin. And there's a history of people using antioxidants to try and prevent some of the lowering of serotonin. And animal studies do support that kind of thing, although my personal feelings on it are that with standard therapeutic doses, you maybe don't have to worry too much about this kind of thing, and that this is more of a concern when people are partying hard, that they might really need to recover.
And then the purity of the drug, we know with ecstasy is often an issue in non-medical settings. So keeping that in mind if people are using ecstasy. Okay, well, we'll wrap it up there. I appreciate your time so much. And everybody who joined today, we'll be sending out the email with the video follow-up and some links that were shared here. And if you want to connect with Entactogen or Dr. Baggott, we'll have that there. But perhaps we'll have to have you come back on and speak specifically about some of these other questions around MDMA neurotoxicity, how we can prevent that, how Entactogen is looking at different novel molecules, which we got a little bit of a taste of, but I know there's a lot more to the story. So thank you so much for being here.
Thanks for having me, and I'd love to come back sometime.
Okay, awesome. Everybody have a great weekend. Take care.